But before we do that, I'd be remiss if I didn't talk about how psilocybin does and does not change the brain. What is and what is not known about that. In fact, when I put out the call for questions about psilocybin, many of the questions related to these issues. The first thing to understand is that a psilocybin journey is really a way to try and put that wedge under the boulder, as I described it, to try and invoke neuroplasticity of a particular kind. And in that way, it's kind of remarkable, if you think about it, that everyone has different lives, different experiences. Psychedelics, in this case psilocybin, are activating these brain networks that each of us has more broadly than they would normally be activated. These are very abnormal patterns of thinking and perceiving and experiencing our emotional and physical life, etc. And yet, so often, the outcomes are positive. Not always, but the outcomes are positive. The experience is positive, even though it might have these anxiety moments or components within them. It's very important to understand that psilocybin and the journey, while important, are not really what all of this is about. It's really about neuroplasticity. So researchers, in particular neuroscientists, are very intensely interested in understanding what sorts of neuroplasticity psilocybin creates. Because it turns out there are lots of different types or processes involved with neuroplasticity. For instance, brain networks, behavior, thinking, emotion, etc., can change because of the addition of new neurons. That's one form of neuroplasticity that's referred to as neurogenesis, the production of new neurons, most typically in the so-called dentate gyrus or other subregions of the hippocampus, a brain area involved in learning and memory. Neurogenesis in other regions of the adult human brain are exceedingly rare, and to be honest, may not occur at all. This is a debated area. We could do an entire episode about this, but for the most part, neuroscientists don't really believe that your neocortex, your striatum, your cerebellum has that much neurogenesis that's related to learning and memory of new things or new experiences. And we don't actually think that occurs as a consequence of taking psilocybin either. Now, some of you who are familiar with, for instance, the cerebellum might be saying, wait, what about granular cell proliferation in the cerebellum? Or what about the rostral migratory stream from the subventricular zone where there are neuroblasts, spitting out little new neurons that migrate into the nose to replenish the olfactory neuron population? Yes, that's all true. That does occur. It's been observed in mice. It's been observed in monkeys. And to some extent, it's been observed in humans. But it's not, again, I repeat, it is not a prominent feature of learning and acquisition of new skills, new ideas, or new emotional states. Perhaps the best supported evidence for neurogenesis underlying new thoughts, experiences, abilities, emotions, etc., is the production of new neurons in that dentate gyrus subregion of the hippocampus. And that probably does occur in humans. But neurogenesis is not really the dominant mode of changing neural circuitry in adult humans. It might be a player in adolescence, in young childhood. It is certainly a player before we are born, when we are still in utero. But then the brain is being wired up in many different ways, including the addition of new neurons and changing of connections. All this is to say that while neurogenesis is a really sticky idea and it makes great headlines, the addition of new neurons is not really the way that the brain changes under psilocybin, other psychedelics, or just generally. It's perhaps responsible for maybe 1% to 2%, and I'm being generous there, of the rewiring events that are going to be most important for all of us. So we need to set that down and kind of cement that there until further evidence comes out to the contrary. That's certainly where and here I feel comfortable speaking for the majority of neuroscientists out there, professional neuroscientists that is, the papers showing adult neurogenesis are interesting, but they don't really explain most of the plasticity that occurs in the adult human brain. So if neurogenesis ain't it, what is? Well, it's very clear that psilocybin, other psychedelics, and any sort of behavioral or drug intervention that can induce neuroplasticity does so largely through the addition or strengthening of new neural connections or through the elimination or weakening of other neural connections. And if you look at the data exploring the mechanistic basis for psilocybin-induced neuroplasticity, it's mostly focused on animal brains, animal models, mice and rats in particular. A little bit on primates, but mostly mice and rats, because that's where the interventions can be done of knockout animals, of imaging the brain in real time. Of course, there are the beautiful studies of Robin Carhart-Harris and others exploring neuroplasticity at the level of brain imaging, at the level of ultrasound measurements of how active are certain brain areas in humans, how extensive is the modularity or not extensive is the modularity, et cetera, the stuff we talked about earlier. So in other words, there are neuroplasticity studies with the effects of psilocybin in humans. But in terms of underlying mechanisms of neuroplasticity, I think the predominant theory is that psilocybin induces neuroplasticity through the addition of novel connections in those pyramidal neurons of the frontal cortex, elsewhere in the cortex, and certainly also in the visual cortex, probably also subcortically as well, below the cerebral cortex, in areas like the thalamus, maybe even in the brainstem as well. And that those neuroplasticity events are structural and functional. And they involve a couple of basic events, the most prominent of which is the growth of dendrites. Dendrites are those little branches or processes that come out of the neurons, not just the pyramidal neurons, but other neurons as well. But since we're talking mainly about pyramidal neurons today, both the apical, those ones that they're called the apical tufts. They're the ones that reach laterally to connect with other neurons, communicate with other neurons that we talked about before, as well as the dendrites that come out of the base of those pyramidal neurons. Those processes grow in response to psilocybin, as well as the addition of what are called dendritic spines. So the dendrites are the branches. The spines are these little protrusions that grow out. They actually, and here, I don't know if this is coincidence or not. Again, I always say I wasn't consulted at the design phase, but these little protrusions actually look like little mushrooms. They have a little stalk and they have a little head, a little spine head. And those little spines, so think of these as like little tiny mushroom appearing. Okay, they aren't actual mushrooms. Okay, the first person that puts in the comments, oh my goodness, I learned today that mushrooms grow out of our neurons when we take magic mushrooms. That is not what I'm saying. What I'm saying is that these little mushroom-shaped protrusions that we're calling dendritic spines do in fact grow out of dendritic branches of neurons when animals ingest psilocybin or are injected with psilocybin, and that those little mushroom-shaped protrusions are the sites of new excitatory connections, new locations for input from other neurons to activate those neurons that have those little mushroom-shaped protrusions.